The presentation by S.Petoukhov and V.Verevkin. Harmonic fractal-like features in epi-chains of genomes of higher and lower organisms. The creators of quantum mechanics, Jordan and Schrödinger, pointed out the main difference between living bodies and inanimate ones. In living bodies, genetic molecules play a dictatorial role. Inanimate objects have no such dictatorial molecules, since they are governed by the average random motion of millions of particles. The study and use of the properties of DNA molecules with their nucleotide sequences, which carry genetic information, are some of the key tasks of modern science. See the history of quantum biology in this article. The report presents our research on informational features and dictatorial influence of DNA heredity molecules on living organisms. They talk about universal hyperbolic rules and algebraic harmony in the informational organization of genomes of higher and lower organisms. Hereditary information is recorded in DNA molecules in the form of long sequences of 4 nucleotides or molecular letters: adenine A, thymine T, cytosine C, and guanine G. These DNA sequences for each genome of many organisms are presented in the open access in the gene bank in a standard form, which gives the impression of a chaotic DNA text of 4 letters with a length in thousands of pages. But as known, the genomic sequences of 4 letters are not at all random. Let us now turn to the universal hyperbolic rules that we have discovered for the genomes of higher and lower organisms. Let's start with an example of the DNA of human chromosome number 1, whose chain contains about 230 million nucleotides A, T, C, and G. Let's first calculate the number of nucleotides A. Then, in the chain of its doublets, the number of all 4 doublets starting with A, that is AA, AT, AC, AG. Then, in the chain of its triplets, the number of all 16 triplets starting with A, that is AAA, AAT, etc. And so on for each of the set of n-plets or oligomers starting with A. Take n is equal to 1, 2, 3 till 20. We get the following 20 numbers. It is striking that all these 20 numbers practically coincide with the hyperbolic sequence, whose numerators is the number of nucleotides A in this DNA. The deviations of real quantities from this model sequence are hundreds of a percent only. Knowing the number of nucleotides A in the genomic DNA text, it is possible to predict with high accuracy the values of 19 other named sums of n-plets or oligomers of length n. Since, as it turns out, there is a cooperative connection between them. This new author's method of analyzing and comparing DNA texts is called the method of oligomer sums. Similar results are obtained in the same chromosome when calculating the total number of n-plets, oligomers, which start with any of the other 3 nucleotides, T, C, or G. The resulting 4 hyperbolic sequences for the human chromosome number 1 differ practically only in the value of their numerators, which are equal to the amounts of corresponding nucleotides. Similar results are obtained for all other human chromosomes. For all chromosomes of all tested eukaryotic genomes and for all tested prokaryotic genomes, see published numeric results in this preprint. Next section, genomes and the harmonic progression. The model hyperbolic sequences serve as mathematical standards for the general phenomenological rules of oligomer sums in eukaryotic and prokaryotic genomes. These hyperbolic sequences differ from each other only in the magnitude of numerators in their expressions. By appropriate scaling of numerators, each of these genomic hyperbolic sequences is transformed into the historically famous harmonic progression. The name harmonic progression comes from its connection with the series of harmonics in music, or length of standing waves in a stretched vibrating string. The harmonic progression was studied by Pythagoras, Leibniz, Newton, Euler, Fourier, Dirichlet, Riemann in relation to different mathematical and physical problems. Now the harmonic progression shows itself unexpectedly in all tested genomes. The harmonic progression has interesting mathematical features which can be useful for developing new classes of models of genetically inherited physiological structures. For example, its term is the harmonic mean of the neighboring terms. Knowing on any two adjacent terms of the harmonic progression, one can recurrently construct all the harmonic progression using the expressions for the harmonic mean. The sums of the first members of the harmonic progression are called harmonic numbers. The generalization of the harmonic series is known as the Riemann zeta function. The series of harmonic numbers serve as the discrete analog of the continuous function of natural logarithm. This in particular connects the harmonic progression with the V.Verevkin logarithmic law, which is the main psychophysical law and dictates information features for all inherited sensory channels, vision, hearing, smell, etc., whose organs, eyes, ears, nose, etc., very differ each other in appearance. It testifies that genetic and different psychophysical levels of inherited biological informatics are structurally intercorrelated on the algebra harmonic basis. The harmonic progression and the series of natural number 1, 2, 3, etc. are projectively identical and are transformed each into other by projective transformations. Since in each of them, the cross ratio, which is the main invariant of projective geometry, for any 4 adjacent members is equal to 4/3. This gives an outlet on the rich topic of non-Euclidean symmetries in genetically inherited biological structures, including in particular a known projective nonlinear changing of proportions of the kinematic scheme of human body in ontogenesis. Next section, DNA epi-chains and hyperbolic rules for oligomer sums. By definition, in any DNA strand having a sequence number at nucleotides 1, 2, 3, 4, etc., epi-chains of different orders K are those subsequences that contain only nucleotides whose numerations differ from each other by fixed natural number, 1 or 2 or 3. For example, an epi-chain of the second order contains nucleotides with numbers 1, 3, 5, 7, etc. An epi-chain of the third order contains nucleotides with numbers 1, 4, 7, etc. An epi-chain of the fifth order contains nucleotides with numbers 1, 51, 101, 151, etc. Each DNA epi-chains of k's order contains k times fewer nucleotides than the DNA strand and has its own arrangements of nucleic bases, A, T, C, and G. But unexpectedly, despite of these differences, oligomer sum sequences of the total amounts of those unplets which start with a nucleotide A or T or C or G are modeled by very similar hyperbolic harmonic-like progressions, both in the complete DNA strands and its epi-chains. See numeric results in the preprints shown in slide. This means that new fractal-like and symmetric features of oligomer cooperative organizations of eukaryotic and prokaryotic genomes are revealed. The described results expand knowledge on fundamental structural peculiarities of molecular genetic systems and on algebraic harmony in living matter. Fractals are connected with the theory of dynamic chaos, which has many applications in engineering technologies. We believe that the discovery of the described new fractal-like properties of DNA text relates to their epi-chains can lead to new ideas in theoretical and application areas. Conclusions. New fractal-like and symmetric features of oligomer cooperative organizations of eukaryotic and prokaryotic genomes are revealed and described, which are connected with the harmonic progression and which support the thought on inherited algebraic harmony in living bodies. Secondly, the oligomer sums method, which is a new method of the comparative analysis in genetics, has confirmed its effectiveness and usefulness by results of analysis of epi-chains of long DNA sequences. Thank you very much for your attention.